online, so welcome to our audience online as well. Just before we begin, some technical details. So if you are watching this online, if you have a question, there will be an opportunity at the end to ask questions. If you could type that in the Q&A, and then I will read it out so everyone here can hear it, and the speaker can answer for you. So before we start, just a few words to introduce the LMB. So the MRC Laboratory of Molecular Biology, or LMB, is where we are. It's a government-funded research institute situated on Addenbrookes campus in Cambridge. And our aim is to understand complex biological problems at all levels, from atoms and molecules to cells and organisms. Our remit is to study difficult long-term problems, and in doing so, contribute knowledge needed to solve key issues in human health and understanding. Over the years, LMB scientists have made many contributions to medicine, both in terms of new understanding and in development of new techniques and technologies. Today, I'm delighted to welcome Matteo Allegretti from the Structural Studies Division to give our talk. Matteo studied biological sciences at the University of Roma Tre, where he also obtained a Master of Biochemistry. Due to his passion for membrane systems and bioenergetics, he moved to the Max Planck Institute of Biophysics in Frankfurt as a PhD student to investigate the structure and function of membrane proteins using electron microscopy. As a postdoc, he joined the European Molecular Biology Laboratory in Heidelberg to investigate the structure of membrane-embedded proteins directly in cells. Here at the LMB, Matthew's group is continuing to use electron microscopy and other imaging methods to investigate the macromolecular basis of nuclear remodeling in health and disease. And today he's going to tell us some more about what they're doing. So thank you, Matteo.
Thanks a lot, Alison. So welcome, everyone. It's a pleasure for me to speak in front of all of you today. And today, the two protagonists of my talk will be human cells, like this in the background of this slide, and this big machine that are called microscopes. So who has seen those in LMB? Raise your hand. Okay, quite a few of you. So today what I will do, I will try to shed the lights a bit on some details of how we study the structure of our cells using those big microscopes, in particular with a focus also on trying to understand some cancer mechanisms. So what are we made of? We are made of cells. 20 to 40 trillions of cells. And these cells all of them are alive. So they have living properties. Some of those properties are here mentioned on the sides. For instance, they are able to reproduce, to duplicate themselves. They change with time, so they're able to evolve. They have a certain organization. Here you see, for instance, a cut-open view of a 3D model of a cell. It's like sort of futuristic cities where you see a lot of different buildings with different colors. And this is sort of gives an organization of the cell. For instance, this is the nucleus here in blue-violet color. And one property of the cells that I'm going to deal with you a bit later is the capability to migrate, so to be motile inside the body. If we look at this 2D view, you can appreciate more the compartments or the buildings, in particular the nucleus. And my talk is mainly focused on this because the nucleus is like the brain of our cells. So it contains all the inherited information in the form of DNA. And here the DNA is stored, protected, and also is copied here. And the nucleus is able to actually receive signals coming from the environment and to use this signal to produce molecular machine that can react to such signaling. And the signaling can be, availability of food, or change in temperature, or an obstacle. And this is naturally fundamental for the cell to survive. So if this looks to you quite a full kind of depiction, actually the cells are packed of molecular machine. There are more than 10 billion proteins and metabolites in each of our cells. And these machines are able to perform all the properties that gives us life. So transporting metabolites, balancing fluids, transmitting signals. And that's why we are all interested in understanding all of these machines in our cells. What are they made of? They're made of 20 components that can be imagined as Lego bricks. And from the combination of these components, you have different molecular machine forming, and each of them is different in structure and different in function. And the estimated number in nature is around 100 trillions, but this is only probably only a few of the ones that could be evolved. And so as a summary, we have the DNA as a double helix in the nucleus that has the information to produce proteins, and these proteins give us life, us for the cells and for us. So the field of structural biology wants to try to understand the molecular details of each of our proteins, because often a faulty protein is linked to disease, like you see, a sick person has a faulty protein. So there is this medical link between proteins and diseases. Classically, what we have done, and this is the most efficient way to study the structure of protein, is to break the cell and take out all the molecular machines and solve their structure. And that's the most efficient way. But in the most recent years, what we are trying to do is to try to study the assembly of proteins directly inside our cells without taking them out, because we think that in this way we really capture them while they are acting in the middle of their function. And the information that we hope to get out of this is that we hope that is useful for natural drug targeting. So now I need to go a bit more in detail on some physics, but don't be scared so much, because what I want to tell you is about sizes. So what are we looking at? We look at cells that are around 10 to 100 micron in size. A micron is a millionth of a meter, or if you prefer, a millimeter divided 1,000 times. And proteins are around 5 to 10 nanometers. And a nanometer is around a billionth of a meter, or if you want, a millimeter divided a million times. So this is the scale that we are looking at. So how can we look at them? The only way is to use particles or radiations that are smaller than those sizes. And that's why we cannot use the visible spectrum. So our eyes and our evolution, our cells are tuned to be able to see particles that are between 400 and 700 nanometers in size. And this is not small enough to see proteins, because to see the object has to scatter this radiation, these rays, and this scattered particle have to then reach our amazing cameras, and then we can see it. So we need to use different radiations. One very well characterized is X-rays. Each of you for sure had a X-ray scan of teeth, of bone, and you can see the fine details of your structure. And X-ray is a good option, but we can go even smaller particles, such as electrons. And electrons are amazing. They are so tiny. The estimated number is between 10 minus 16 to 10 minus 18 meters, so billionth of a meter. And we use naturally microscopes that can shoot electrons and a magnifying lens to see and enlarge proteins. It's no way that we can study them at high details. So what I'm going to give you now is a brief overview of how we go from cells to protein structure. Practically, we grow the cells, we are able to cultivate them, we make them adhere to a small metallic grid, around 3 millimeters in size, we freeze them, we keep everything frozen, we pass through a couple of microscopes, we get images on our amazing cameras, we use computer software, for instance, Reliant, that was developed here, and we get structure. It seems easy, but actually it's not. It takes lots and lots of trainings from our sides, months and months of trainings to be able to do this pipeline. But I'm going to give you a bit more detail of what's really happening inside the microscopes with some videos. So what we have is our grid with our cells. And that we grow on the grids. And then we have a machine, a robot, that is able to plunge them in a very cold liquid. So first of all, you will see two pieces of papers that are going to blot away some excess of liquid. And then, like a guillotine, this device will plunge the grid with our cells into a liquid that is around minus 180 degrees Celsius. And now we have our cells that are completely frozen. And we can go from this step into the electron microscopes. So you will see now the grid will go into the first microscope. This microscope is our knife, is our cutting machine. And you will see how it will operate. So the grids are coming in through a shuttle. Now it's coming from the right. And here there are a couple of our grids. And you have two cones. One will shoot electrons, and one will shoot ions. So this one is used to image, to see what's on the grid, and this one is the knife. So this will shoot ions that are very strong and will deplete material. And why do we want to do that? You will see subtly here. Oops. And here you have our grid, and these droplets are human cells. And what are going to happen is that we are in this chamber, and from two rectangles will appear, and one will start to deplete material from the top, and one, number two, from the bottom. And we will get closer, closer, and closer until we hit the cell, and we are going to make a very thin slice of this cell. And this happens stepwise, in an automated way, for dozens and dozens of cells. And you are going, here you see, this is getting thinner, and then again thinner and thinner. And when we have thin enough slice of our cell where the electrons can pass through, we are good to go to the next microscope. That is the one that will give us images of the content. So we have our thin slice, and we do it several times in an automated way. And then we take the grid, we go to another giant fridge, keeping everything cold. And what's happening here is that we use the electrons to take images of our cells at different orientations. And here you see the images coming out. Because only if we get the images at different orientation, we can reconstruct volumes. We have all the information to reconstruct volumes. And those volumes contain high details of protein and membranes that are in our cells. And this is the information that we want. I show you another example here, where you have actually how the images come from. The color is just a rendering. Here, actually, the images come all in black and white. You see some nuclear membrane and proteins that are some annotated here. And what we do, we collect hundreds of those, and then we solve structures. But the important thing is that we can solve structure in different states. For instance, here, you see the structure of the gate of the nucleus, the gate of our brain in the cell. It opens and closes according to the energy status of the cell. And this is beautiful. And this we cannot do if we would have done the extraction of proteins from the cell. So we need to be inside the cell to see changes according to the physiological state. So what my lab is studying, we are studying how the nucleus is changing in several processes at the beginning of human life in the form of sperm and fertilized eggs, and also in cells that are moving. So they are both very motile cells. And they are moving to look for a pathogen. They are moving because they look for nutrients. Or, unfortunately, there are also cells that move in a not controlled way and in an unwanted way. And these are cancer cells that metastasize, so they go away from their origin and they go in the blood. They pass through very tiny constrictions and they spread all over the body. Naturally, this is lethal. And the question that we are trying to answer is, how can a nucleus, this around 5 to 8 micron in size, be squeezed to this kind of constriction? You know, the nucleus, I said, is the brain and contains the DNA. How can be not damaged during these processes, and why the cell is not dying? And that's the question that we have. So how can we study this in our lab? Well, there are ways to do this, because we cannot do this in our body. So we do it in the lab, for instance, using plastic channels. And here in green, you see the nucleus. And you see that this image shows the nucleus managing to pass through this small constriction, but there are no molecular details. How are the molecular machine allowing this process? And that's the question that we have. So we want to use our cells, growth on grid, with specialized grids, to understand, using our electron microscopes, all the molecular machines that allow the nucleus to pass through this migration and constricted places. And this is the summary. So we investigate proteins that allow the reshaping of the nucleus during the migration of cells in healthy cells and in sick cells or cancer cells. And we use the microscopes that I show you and computer software to solve the structure of such proteins inside the cells. And we hope that the structure will be useful for developing new tools against cancer. Here we are. This is the end. We sit naturally at the floor in the north side of the LMB. And the group, the amazing member of the group, Alia, Piotr, Oliver, Tom, and Vicky, and all our fantastic collaborators. And thanks a lot to all the staff, because you allow us to work smoothly, and without your support we couldn't do what I presented today. Thanks a lot.